Welcome to Eduimagica, the medical mystery education hub. Today we are going to learn about Krebs cycle or the citric acid cycle. The Krebs cycle is also known as citric acid cycle, tricarboxylic acid cycle or Saint-Georgi-Krebs cycle. The Krebs cycle is a biochemical pathway that is used to generate energy through oxidation of acetyl-CoA. Krebs cycle is also used for generation of NADH and production of amino acids. The citric acid cycle is a closed loop. The last part of the pathway reforms the molecule used in the first step. The citric acid cycle includes 8 major steps. The citric acid cycle takes place in the mitochondria of the eukaryotes and the cytosol of the prokaryotes. Now let's talk about different steps involved in Krebs cycle. So the very first step involved is a preparatory step, which begins from pyruvate. Pyruvate is derived through glycolysis of glucose, in which one molecule of glucose splits into 2 molecules of pyruvate. About glycolysis, we have a separate video, so you can find the link in the description below. Now this is a very important concept, please pay attention. Now the question arises how pyruvate from glycolysis is converted to acetyl-CoA so it can enter the citric acid cycle. So the answer is through pyruvate oxidation. Pyruvate is produced by glycolysis in the cytoplasm, but pyruvate oxidation takes place in the mitochondrial matrix. So before the chemical reactions can begin, pyruvate must enter the mitochondrion, crossing its inner membrane and arriving at the matrix. Overall, pyruvate oxidation converts pyruvate, which is a 3-carbon molecule, into acetyl-CoA, which is a 2-carbon molecule attached to coenzyme A, producing an NADH and releasing one carbon dioxide molecule. In matrix, pyruvate is modified in a series of steps. In the very first step, a carboxyl group is snipped off or removed from pyruvate and a carbon dioxide molecule is released, leaving behind a 2-carbon molecule. And in the next step, NAD+ is reduced to NADH. Now moving on to the third step. In the third step, an acetyl group is transferred to coenzyme A, and coenzyme A is an organic molecule derived from vitamin B5 to form acetyl-CoA. The steps above are carried out by a large enzyme complex called Pyruvate Dehydrogenase. Now this is also a very important concept. Now the question arises why we make acetyl-CoA. So the answer is acetyl-CoA serves as a fuel for Krebs cycle in the next stage of cellular respiration. And the addition of CoA helps activate the acetyl group, preparing it to undergo the necessary reaction that takes place in the Krebs cycle. Now let's move on to the different steps involved in Krebs cycle. In the very first step, the citric acid cycle, the acetyl-CoA joins with a 4-carbon molecule oxaloacetate, releasing the coenzyme A group and forming a 6-carbon molecule citrate. And this reaction is catalyzed by the enzyme citrate synthase. Now in the second step, citrate is converted into its isomer, isocitrate. This is actually a 2-step process involving first the removal and then the addition of water molecule, which is why the citric acid cycle is sometimes described as having 9 steps rather than 8 steps. Now in the third step, isocitrate is oxidized and releases a molecule of carbon dioxide, leaving behind a 5-carbon molecule, that is alpha-ketoglutarate. During this step, NAD+ is reduced to form NADH. The enzyme catalyzing this step is Isocitrate Dehydrogenase. This is important enzyme because it is used in regulating the speed of citric acid cycle. The fourth step is similar to the third. In this, alpha-ketoglutarate splits into succinyl-CoA with the help of an enzyme alpha-ketoglutarate dehydrogenase. And in this reaction also, one molecule of NAD+ is reduced to NADH, releasing a molecule of carbon dioxide in the process. the remaining 4 carbon molecule takes up coenzyme A. This is also a very important enzyme in regulation of the citric acid cycle. In the step number 5, the coenzyme A of acetyl-CoA is replaced by a phosphate group, which is then transferred to ADP to make ATP. In some cells, GDP, that is guanine diphosphate, is used instead of ADP, forming GTP, guanine triphosphate, as a product. The 4 carbon molecule produced in this step is called succinate, and this step is catalyzed by the enzyme succinyl-CoA synthase. Now in the step 6, succinate is oxidized, forming another 4 carbon molecule called fumarate. In this reaction, 2 hydrogen atoms with their electrons are transferred to FAD, producing FADH2. The enzyme that carries out this step is embedded in the inner membrane of mitochondria, so FADH2 can transfer its electron directly into the electron transport chain. Now moving on to the next step, in the step 7, water is added to the 4 carbon molecule fumarate, converting it to another 4 carbon molecule malate, and this step is catalyzed by the enzyme fumarase. In the last step of citric acid cycle, oxaloacetate, the starting 4 carbon compound is regenerated by the oxidation of malate. Another molecule of NAD+ is reduced to NADH in this process. And as we all know, this citric acid cycle is a closed loop cycle. Now let's take a step back and do some accounting. Let's calculate the yield of the Krebs cycle. Overall, 1 ton of the citric acid cycle gives 3 molecules of NADH2, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. But as we all know that glucose gets split into 2 molecules of pyruvate, and 2 molecules of pyruvate splits into 2 molecules of acetyl-CoA. So this needs to be doubled. So the yield we get is 6 molecules of NADH2, 2 molecules of FADH2, 2 molecules of GTP, and 4 molecules of carbon dioxide. And at last, the yield produced in the Krebs cycle is then transferred to electron transport chain for the further production of ATP, as Krebs cycle cannot generate the ATP directly. Thank you for watching. You can follow us on Instagram, Facebook, and Twitter. The links are in description below. You can also visit us at www.eduimagica.com. Please do subscribe and hit the bell icon.